Hi, my name is Frederico Azevedo. I'm a postdoc at the Center for Brains, Minds and Machines, and I would like to welcome you all to our conversation with Eric Shea-Brown, a professor in the Department of Applied Mathematics in the University of Washington. Welcome to our center, Eric.
Thanks so much for having me, Fred. It's a real privilege to be here. I'm looking forward to talking with you.
Could you tell us a little bit about your scientific background and your research interests?
Sure. I think like many people of my generation, everything started as a teenager with reading Stephen Hawking and A Brief History of Time, and we fell in love with the idea of physics and the idea that there are these fantastic predictive rules that can be stated compactly, and suddenly the universe unfolds from that. So I went to college to study physics, and I remember having an amazing moment in classical mechanics class. These are very basic things, but for me they were astonishing when somebody used a geometrical picture, I think it was a phase plane for a pendulum, to try to understand the way something evolved dynamically over time, and I thought I was in paradise, some sort of unity of geometry and dynamics. And then I had this very open-minded and kind dean of my program who encouraged me to explore other options for an experimental lab class, and I ended up finding my way into an experimental neuroscience lab, an electrophysiology lab, which blew my mind, and I'm grateful to the gracious people who took a naive young person like me into that environment. And then the kicker came, which is I got to take a biophysical neuroscience class from somebody named Harold Lacar, and there it was on the board, the same geometrical picture, the same phase plane, and this time explaining a nonlinear oscillation in the, the Morris-Lacar, after Harold Lacar, single neuron model. And suddenly I knew that I could have my cake and eat it too, study sort of modern, new, and completely open questions, unlike physics, in which many of the sort of astonishing discoveries were well underway from a mathematical point of view, to try to work on a different type of new field, but using that type of tool and that type of mathematical and theoretical surprise. I didn't know that was a choice for me, so I just got lucky, thanks to generous people, and that kicked things off. Anyway, so that was undergrad. Then I looked for a place to do grad school at the interface between dynamical systems, which is a branch of math that you can tell that I love, and neuroscience. I found a wonderful once again, very generous and giving pair of advisors at Princeton. Was Phil Holmes, who's a dynamicist, and Jonathan Cohen, who's a cognitive neuroscientist, and they mentored me to the present day through a wonderful period as a postdoc at NYU with John Rinzel, yet another pioneer of the interface between dynamics and neuroscience. So good luck and good people. That's how I ended up in the chair.
And what are you doing right now in your lab? Which is the main area that you're researching?
Yeah, a mixture. I've got to say we're struggling with trying to bring together bottom-up models of neural dynamics, the type of thing that we were just discussing, right? Put together a bunch of cells. They each have their predictive models for how they transform inputs into outputs, connections, likewise, get some sort of collective picture of the dynamics of a network of these things. Bottom-up approach, right? Standard classical, maybe physics style, and these top-down approaches, right, that a lot of your colleagues here at MIT and really around the world are pioneering. This is new to us, but once again, you get some sort of a dynamical system out of a neural network, but this comes from a particular objective, right? A top-down objective from which a system is trained. So how are we going to bring these two perspectives together? We are absolutely not novel. We're probably not pioneers in this area, but we are fascinated, and we're joining people really, again, around the world and trying to bring these two angles together. And we're struggling a lot of the time, taking a lot of wrong turns, but having a good time.
It's interesting because our brains, they have tens of billions of neurons.
Yeah.
And each individual neuron can be connected to hundreds or thousands of other neurons. So imagine now that we have a very advanced technology that can give us the electroactivity of each single neuron with a very high temporal resolution, let's say. How would you start analyzing such data in order to understand the general principles of how the brain works?
Fred, you are a tough interviewer.
This
is exactly the question that you would expect on a job interview, right? And after that, you have to tell me, where is my lab going to be in 10 years? And then what's my next big NIH grant? It's a great question, and this is one we all have to get really serious about now. You're absolutely right. This used to be kind of a playtime thought experiment question. We'd have it over a sandwich or a beer, and then we'd return back to the sort of limited resolution at which we were doing our theory or at which we were collaborating with our experimental colleagues. That's not true anymore, right? We really need to have a big rethinking of our approach as theoreticians, as applied mathematicians, and as neuroscientists to meet the point which you're describing, because it's coming, right? In some ways it's already there for simple model organisms, and we'll probably be getting close enough to that point soon, maybe in some basic mammalian systems or mammalian brain areas, certainly in our lifetimes, Fred. Well, what are we going to do? What's the place to start? Well, there's a group of people, Manish Sahani and I have had this discussion. Others, I'm sure, have independently had the same type of ideas. What if there's a meeting point, right? Again, between dynamical models or the levels at which we can pose theories, really in terms of mechanical equations or mathematical equations. What if there's a meeting point between those theories and this very high dimensional data that is in terms of really the effective dynamics, a lower dimensional approximation of the system that can be identified? Can we put down the right meeting point between theories and these very high dimensional data that discards a lot of the features of the data, but not more than is hopefully necessary? So I think there's an important point here. We might say, oh, we're going to do some sort of reductionist modeling based on these hundreds of millions of cells. We're going to try to follow the lead of others in the field and do some dimension reduction, again, identification of rule-based dynamics that evolve in a lower dimensional space. Maybe we're tossing the baby out with the bathwater. Probably not true, right? There will be some level of resolution that the recordings or the experiments need to have in order to get the right reduced model. So the hope is that that's what we can do, right? That type of reduced data-driven, I think dynamical systems modeling, and others in the field are also, again, working in this direction, apply the same techniques to ground up or more mechanistic models and hope to find a meeting point. But you can see, right, this goes back to, is this just a chat over beer and then you just go back to what you're doing or not? I think these are opportunities, and again, many recognize this right now for experimental analysis, even though we're not all the way yet to these huge high dimensional recordings, and for theoretical analysis of model systems to see how much we need in terms of numbers of elements and dimensionality of data to derive and really stably derive a class of effective dynamics, and have that class of effective dynamics or class of effective models be invariant to adding more and more.
So you think that with the current technologies that we have nowadays, we are able to isolate the right summary statistics in order to predict the collective behavior of some brain networks?
Well, that's a great question. And again, I think that it's really going to be dependent on the question and what we really mean by prediction, right? Many know famous work, for example, I just pulled this up, I'm not that familiar with it, but I know it at the pop sci level of Jack Gallant at Berkeley, right, who was able to decode many aspects of visual movies from extremely coarse spatiotemporal measurements from the human brain, fMRI style measurements. Well, in order to predict then what it is that the brain is representing, at least on that level, we've had the technologies for decades. For other features, probably involving the mechanics that enable the system to learn quickly from examples or switch contexts, or maybe most certainly the mechanics that describe some types of dysfunction that are known to have, or pathologies, neuropathologies that are known to have origins at cellular or even molecular levels. For that type of question, we are going to need to be able to drill down to a much higher levels of resolution. But it seems like we're kind of already out of the gate with examples like that, and other examples perhaps that are limited in terms of behavioral complexity.
Actually, this brings us exactly to the next question, is that In order to understand the dynamics of communication in complex brain networks, several groups, they decided to study the brain at different levels. For instance, you have groups studying the synaptic activity at the molecular level, and then you have other groups of people who study electrophysiological recordings from single neurons.
Yeah.
And then you had other groups that study the brain at the millimeter scales, for instance, with fMRI, which measures the indirect activity of neurons, the population of neurons which are confined in a voxel. So the question is, if you want to understand complex behavior such as a cognition, how much detail do you need to collect?
I love it. I don't know. And I mean, an easy way out is to say that, it's going to depend on the question and repeat the answer that I had to your previous line of conversation. But another angle on that might be to think about evolution. I'm no expert in this. You already know my background, but we certainly know that a lot of complex behavior in terms of goal-directed sensing and movement arose well before we had large-scale anything, right? Really molecular interaction networks at the beginning. And a lot of those same sort of multifaceted molecular signaling compounds persist, right, in our own genomes as primates. The probability that somehow those are no longer important, given the level of conservation, is low. And so what we need to do, of course, is a bit of a buzzword, not mine at all. On the one hand, multi-scale type of modeling to see what the reflection of these molecular type of mechanisms are at higher levels, but of course also functional experiments in which we manipulate these much finer scale markers and see what the impacts are on cognition. Maybe another angle on this, and again, others I'm sure are pushing in this direction, there's the disease point of view, right, where we see that at least the initial signatures in many unfortunate pathologies are expressed at the molecular level. So we know that these, if we really want to understand the brain and be helpful to society in that one avenue in which we can contribute, right, we have to go clearly to that level as well. So the small scale stuff really has to matter, and we have to embrace it, and bridging to the level at which we know that we can sometimes read out behavior and predict behavior is one of the reasons why we're so lucky to get to do this for our careers. It's such a beautiful and spectacularly hard problem.
Changing now the line a little bit, it's clear that the transmission of information in the brain depends on neurosubstrates. But several studies have shown that actually the functional and structural connectivity aren't necessarily tightly linked. So in your opinion, how much does the functional behavior of brain networks depend or are related to the structural connectivity?
Oh, fantastic question. I guess there are a couple of different avenues that'll determine that question. Let's start with maybe very known circuitry in very early visual system, for example, like the retina, right? So in that case, there are aspects of functional connectivity that drive what we think of as the result of connectivity, namely synchronized or correlated activity among pairs of cells that matters for function, okay? So that's functional connectivity giving you functional sort of collective dynamics in a way that matters for the encoding of signals that can be directly traced to divergences in, for example, the pathways that lead to particular parasol cells, is the work of Frederico and colleagues. So there are cases in which the neural substrate for not just functional connectivity, but the type of functional connectivity that actually matters for computation can be traced directly back to the circuitry. Then, as you rightly say, there are situations in which a given level of structural connectivity seems to either matter or not, perhaps due to modulatory conditions, perhaps due, as Eve Martyrs and others have suggested in beautiful opinion pieces backed up by really very concrete electrophysiology data, where you may have a fixed structural level of connectivity, but what that actually means for function changes due to the cell's local dynamics in which they might for example, ignore inhibitory inputs for one state of adaptation or regulation of the ion channels in a particular cell. They might ignore this inhibitory input, it just switches me off, but if you have activation of some sort of rebound current, then suddenly you become sensitive to an inhibitory connection in terms of driving firing that isn't there before. Okay, so in the retina, as I started out, we don't have, as far, at least in the simple example that I mentioned, at least there is a behaviorally relevant condition in which the structural connectivity maps onto the functional connectivity. In the cases such as Eve Marder and others have studied, just look up her work on functional connectivity, you'll see it immediately to the audience, I guess. You have that the structural connectivity does matter, but it gets mapped into a discrete set, perhaps, or a continuous family of different functional connectivities in ways that we can trace based on understanding the nonlinear dynamics of the circuitry. And what is more interesting than that is sort of functional reconfiguration of circuits. Then the other possibility is that the structural two other possibilities. One is that the structural connectivity that we are actually observing does not map onto the functional, actually I'll wrap this into one point. So the third possibility, I think, is that the functional connectivity that we're observing does not map onto the structural connectivity that we're comparing it with. And two possible reasons for that are simply that there are hidden units or hidden nodes in the connected network that we're simply not observing. We're observing a subset of cells, that's it. Some EM experiment, for example, electron microscopy experiment, we might be imaging only a subset of the cells, and then there will be long distance processes that we're not resolving. So it could be that there are really hidden units that we're not resolving structurally, so we just don't have the right structural connectivity. And then the other possibility, of course, is that there's co-modulation due to either common sensory signals or co-fluctuations in behavioral or modulatory state that are driving large amounts of apparent synchrony that are regulated by connectivity, but it's sort of global connectivity to a signal that might not even be transmitted through the traditional synapses that we're using to define the structural connectivity. So in the end, I know it's not a magic system, right? You know, it's a bunch of electrophysiological devices, but really defining what the structural connectivity is that drives this system, I think is a big challenge for us. But I'll go out on a limb and be optimistic and say when we have the right level of observation and when we embrace the right ways in which the circuit can reconfigure itself to pay attention to or ignore different types of interactions between the cells, I think we'll see a coming together and an understanding of functional connectivity in terms of structure.
Yeah, there's definitely a lot to do.
Yeah.
As the last question, I'm sure that many students are going to be watching this video later and they will be very inspired by your research. So which piece of advice would you give to an undergrad that will be applying to a PhD program very soon and wants to understand how the brain works as a whole? Which areas should they focus on, and is there an ideal balance between theory and experiments?
That's so kind of you to say that and to ask. Doesn't it feel like the landscape's changing, Fred, so fast? You look a little younger than me, that's a compliment, but I'm 40 or so, so when I was in that position 20 years ago, I wasn't aware of, maybe I was ignorant, but I wasn't aware of really career tracks, stable career tracks at the interface of theory and experiment, and really vibrant theory communities that existed within neuroscience communities. Maybe I should have come here and visited this spectacular enterprise at MIT and been proven wrong. But that space for doing theory and really having a community of colleagues, fellow students, and faculty within a biology environment, that is something that one would naturally expect to see as the field becomes more quantitative and as theories become more successful. That's happened in every other branch of science, and that's going to happen over our lifetimes and over the lifetimes of those young students. So hoping to find a place in which you're having your cake and eating it too, but doing so with a bunch of other colleagues in a vibrant environment, I think that's a new option. And so looking for those communities is the first thing that I would say, because there's something about this field. We don't all do the same thing. We don't all know theoretical neuroscience I'm discussing. We don't all know the same type of mathematics. I'm very weak, for example, in some areas of machine learning and computer science. But even so, I'm so interested in this stuff, and I'm not unique in that way, that when I get in the room with somebody who's attacking the brain using theory, I really want to know what they have to say, and it's just exciting, and it feels so good. I think the same is true for others, maybe from those types of backgrounds who interact with somebody who's trained more in mechanistic modeling, like me, but who's open to the other point of view. So I guess advice point number one is that these communities of people using theory to study the brain now exist in more and more places, and they actually exist in places which, as a young mathematician, so you might not expect to find them. The other piece is the same thing that probably everybody else says all the time, but that's to track down the real joy and excitement in problems. And that's another thing that I think is maybe a little bit new, is that the problems that as a young theorist you might think are really, really exciting are also those that are increasingly mainstream in neuroscience and in systems neuroscience. So if you have maybe my talk will get panned today, but if you have some kind of abstract question, let's just pose it out there, how does dimensionality in a neural system depend on some feature of its connectivity or the tasks on which a system is trained? It's kind of abstract, it's not totally exotic, but you can talk about those things and engage very directly with those things with all kinds of people across the field now. It doesn't matter what their background is. And so I think following your passion as a young theorist to the areas where you just feel excited by the mathematics and by the openness of the problems is really the opposite of a dead end in terms of really interacting with the larger and well-funded and job-having neurobiology community. I think that's something that's new.
So in summary, you should come to the CBMM.
Hey,
right?
Okay, it looks like a good deal so far. Yeah, thanks a lot for having me. Great conversation.
Thank you very much for having been here. Eric is going to be giving a talk later at MIT today, and this talk is going to be online for all of you that want to watch it. And I thank you all for watching us, and see you next time.
Thanks a lot for having me.